Well, thanks for the intro, Jean-Baptiste. Yeah, I'm Jacob. I work at INRIA, which is in the Paris region. And just for some context, I originally come from a background in mathematics, but throughout my career, I've drifted quite a bit in the direction of biology. I've also been a little bit in the direction of control. At least I got my PhD from a control engineering lab. So um, I do have some interest in that direction. But today, the focus of the presentation is going to be on some work that we've done on models of stochastic reaction networks and how to couple them to dynamics that occur in the context of growing cell populations or microbial consortia. So the context in which I'm going to present you this is going to be synthetic biology. So that's sort of doesn't necessarily mean that what I'm going to present is limited to being useful for synthetic biology. But it so happens that all of the collaborators with which I've been working in biology are working on synthetic biology applications, and also the grants that we've written and submitted are for those applications. So it's good enough motivation for pitching this work. So just, I guess for many of you, this will be pretty unnecessary to introduce, but the original vision of synthetic biology was that we can create biological circuits or systems sort of akin to how one constructs electronic circuits by going from parts to devices and systems with genes and proteins playing the role of parts that are assembled into gene circuits, which are then placed into cells where they carry out the function for which they were designed. Now, the problem with synthetic biology is that if you take a gene circuit and you place it in a real living cell, then in the majority of cases, it's not going to do what you thought it would do, not going to do what it was designed to do. And there's a multitude of reasons for this. A reason that is going to be of interest, or that's always of interest to me, is stochasticity of the circuit, because these circuits are all based on chemical reactions inside cells, which may be stochastic. But there are also other, sometimes more important reasons, such as interactions that the circuit may have with natural processes of the host cell. For instance, the circuit may use up some resources that the cell has, and then that the cell cannot use it anymore for other tasks that it does, such as growth. So this may create a burden for the cell and can lead to all sorts of complicated consequences that were not really predictable at the onset. But this is sort of the classical problem or the wall that synthetic biology has run into in the past. And there's been a lot of work that tried to cope with that in some way, like modularity in the designs and all those sorts of things. Among the things that people have been doing, there's also what's called cybergenetics these days. So that's sort of more going into a control direction. And you say you could interface the circuit that you construct with some feedback control systems that robustify its behavior, or that stabilize the dynamics or make it immune to perturbations, and so on and so forth. You can try to design control circuits that operate inside single cells, or you can go via an external feedback loop if you have a measurable output of your system as well as an input, such as a chemical inducer or an optogenetic signal that you can tune from the outside. So I've been doing quite some work in that direction in the past, but it's also not really what I want to focus on in today's presentation. What I'd like to focus on is where sort of this vision here still falls short. And that is that, in fact, the mindset that is sort of presented in this slide is that the system for which we're constructing our gene circuits is a single cell. But that's not really matching what we want in the majority of applications that we have. Because eventually, if we do, say, synthetic biology for bioproduction or something like this, then we're not going to care about what any single cell does. We're going to care eventually what the full population of growing cells or a microbial consortium is doing. And that means that the output of interest here that we have is actually something that is defined at the scale of a growing population. So that is something that we then would need to take into account if we think about how we actually design the single cell circuit to optimize its function. So the talk that I'm going to do today is on the one hand on sort of I first spend a bit of time trying to convince you that it's really important to sort of change the mindset that we have from the single cell to the population scale. And I'm going to do that by shooting a bunch of examples at you that I've been confronted with myself, where these things are, so these couplings are, or the population scale is important. And then I'm going to move over to sort of introduce a modeling framework that we can use to actually study sort of joint dynamics of the single cell systems that we are constructing and the dynamics of the growing population. So I'm going to start shooting examples now. So this is a pretty trivial sort of classical thing that people are doing in or have been doing in synthetic biology already sort of decades ago, or 1 or 2 decades ago, I guess. So this is this synthetic circuit here is a NOT gate. So that's a particular circuit that has been constructed by a collaborator, Lorenzo Pozzotti. So a NOT gate, so there's an input here, which is a chemical inducer that's called HSL. It activates a gene, which is TET-R here, which represses a fluorescent protein, which is the output. So that's a NOT gate because the output is present when the input is absent, and the output is absent when the input is present. So then the output sort of reports in the NOT way on presence or absence of the input. And people have been describing these sort of circuits with Boolean models in the early days, I guess. And maybe you pick up a bunch of those gates and you put them together to form more complex synthetic circuits. But this is really this thinking about that as sort of a Boolean thing, a switch with a 0-1 output is really falling short on really telling you what's going on when you take that circuit and you place it into a growing population of cells. So for example, what you see on the bottom here is some data that one can measure of this particular circuit. And bottom left, what you'll see is a constitutive gene expression in cells of that population. So there's an constitutively expressed GFP here that's expressed in those cells, which is completely independent of the circuit. But you're going to see that as a function of increasing levels of the input, which is on the x-axis here, you get some sort of weird consequences for constitutive gene expression levels in the population. So that means that this circuit really interfaces with just other gene expression things in those cells. And another thing you can look at here is actually the net population growth rate of cells or cell populations carrying that circuit. And you'll actually find that the growth rate decreases as you increase the input here, which is because the production of some of the proteins in the circuit is actually burdensome for the cells, and it reduces their growth rate, which creates all sorts of weird consequences and feedbacks on the circuit itself. So the bottom line here is with this logic gate mindset, we're not ever going to be able to explain data such as what we've measured here. We need really something that describes the single cell circuit, but that also takes into account somehow what the circuit has as a consequence in the context of a growing population. I'm going to continue shooting examples. This is another one that I've spent a lot of time on. That's a circuit that has been constructed by a former PhD student, Chetan Aditya. It's now a postdoc in the group of Jared Thatcher at Princeton. So he did that work as a PhD student in the lab at Pasteur when him and me were both still working at Pasteur in the lab of Gregory Batt. So what he wanted to do is he wanted to create a synthetic circuit that would allow us to create simple microbial consortia of different cell types. So actually try to make a cell type switch as a synthetic circuit. And the way that he did that was that he used first an optogenetic system. Some of you may maybe know that from a lot of work of the Kamash group on this. So there's an EL222 optogenetic system here. So EL222 transcription factor constitutively expressed. And when you shine blue light on the cells, transcription factor dimerizes and activates the production of Cre recombinase, which is the yellow thing here. So this recombinase then recognizes a piece of DNA on the cells, and it cuts out that piece of DNA. And these cells here, they are designed such that when this piece of DNA is cut out, you remove a blue fluorescent protein from the cells. So that's previously that is produced, so the cells are blue. But then when you remove it, they stop producing the blue, and instead that moves a green fluorescent protein downstream of that promoter here. So effectively, these cells, when they recombine, they switch from blue to green. And of course, this is constructed for us such that we can actually observe the recombination event in cells. So the sort of practical application of this is that you can create division of labor, a simple microbial consortia here, where you can have two different cell types. You can trigger a flip with an external light application from one cell type to the other, and then you can design the different cell types in the way that you like to sort of each carry out a different function in your population. So all the rest of this slide is basically just showing that the system is working. So I'll just take a look at the B panel here very briefly, and then I'll continue. So what you see in this one here is this is actually flow cytometry data. Time going from the top to the bottom, and this is showing green fluorescent levels of cell populations that have been exposed to blue light at time 0. So before they were in the darkness, because they were in the dark, they have all not any green fluorescent proteins, so they had low levels in green fluorescence, so they're all this blue type here. And then as time progresses, these distributions shift to the right, and eventually you get the entire population to be bright green fluorescent and of the recombined type. You can look at this under the microscope by flow cytometry, dynamically, in whatever way you like. It's a very nice system that works very nicely. I could tell lots of stories about it, but I really want to just like zoom in on one particular set of experiments that we've been doing with this. And that's actually shown here. So what you see here is a set of experiments that have been carried out. So this is with cells that are growing in bioreactors. So there's always growing populations of cells here. And each of these set of colored dots in the plot on the left, they correspond to one experiment. And the difference between all those experiments is that cell populations have been exposed to blue light for different durations of time. So for example, these black dots here, this is a population that has been exposed to no blue light at all, so they're just kept in the dark. And then we can see over time, you get 0% of the population to turn from the blue to the green type, which is what's shown on the y-axis here. So you increase the duration of the light that you apply. So, for example, this one here is cells are exposed to blue light for 15 minutes, and then they are put back to the darkness. Then you get something like approximately 20% of the population to turn to the green type. And there are two things in this which I'd like to point out, and one is pretty cool and the other is pretty curious. So first, the pretty cool thing is that what this actually means is that we can vary the duration of the light that we expose our population to, so as to regulate the fraction of the population that we're going to turn to the new cell type. And that's really what gives us, a handle or controllability here on the population composition with the external light input that we're applying. So that's the cool thing. This is really what makes the system do what we want it to do. And the curious thing is that these cells are growing in bioreactors that are well steered, that are uniformly exposed to light. So what this actually means is that in a case where, for example, we get half of the population to turn to the new type, we get half of the cells to respond to the light and half of the cells to not respond to the light, despite the fact that these cells are all genetically identical and exposed to the same light and so on and so forth. So there's really like stochasticity in the response of the cells, or variability between the cells in the population. that leads to different decisions that they're making here and whether to recombine or not. And in fact, it's exactly this stochasticity in the system which makes it possible for us to control the composition of the population. So it, by design, sort of this system operates on stochasticity and cell-to-cell variability. What I'd really like to point out from this system is to link that back to my motivating introduction in the beginning. So what we're dealing with here is again a situation where we've designed and constructed a single cell circuit. So here it's this recombination system. In fact has stochastic dynamics, which are of core importance to its functionality. But eventually, what we care about is an output which is defined at the population scale, like I motivated in the beginning, right? What we want to do with this is we want to control the composition of the population. So if we would want to use a model here to quantify and describe that system, maybe to have a model that helps us control dynamics here, we're going to need to write something down that tells us how the single cell circuit responds to light, but then is capable of mapping that into the emergent behavior that we get at the population scale. So again, this example is not the one that I'm going to use further in this talk. I'm going to refer you at the papers that we published on it. They have been out for some time already. I'm gonna shoot a third example at you now, which is the most trivial of all examples in some sense. And that's the one that I'm going to use as sort of, I'm going to pick up again as running example in the talk. And this is in fact an example which I like very much because it couldn't be more trivial. So it's so trivial that basically any lab on this planet could carry out the experiments that I'm going to show you with this. At the same time, it's a problem that's so widespread that it's almost present in, I don't know, I'm not the expert to judge that, but maybe, I don't know, half of synthetic biology studies or so. And still, like, at least from my perspective, I feel like it is actually a system that's not all that well understood. And this, you can already see it in the slide. I'm talking about plasmids and plasmid copy number fluctuations here. And in fact, the plasmid that I'll be talking about here is an absolutely trivial plasmid. It's something that also Chetan constructed and used. It's a 2-micron plasmid. We're doing experiments in yeast here, just for context, but it's not all that important. And what I want to focus on this plasmid is really almost nothing. There's nothing more here than constitutive expression of a fluorescent protein from that plasmid and no more. Except that, of course, there's also a selection marker, because there's always a selection marker on plasmids in synthetic biology, because if you don't put a selection marker, then your cells will not, or your population will not maintain that plasmid. And that's actually important. So what does that mean? So here in this case, the selection marker is an essential amino acid. So from the plasmids, the cell can produce an essential amino acid. And then usually when you grow your cell population, you grow it in media that does not contain this amino acid, which means that cells that don't have the plasmid, they lack the amino acid and they are not viable. So they're selected against. And that ensures that the plasmid is maintained in the population. And sometimes, so in many cases, like biologists would be content with this, right? You just put a selection marker, then you use selective media, and then you forget about the fact that you ever did this. Sometimes you may also want to go a bit further and say, now I'm going to study the stability with which my plasmid is actually maintained in the population when you're not using selection, right? And a way in which you can do that is by doing a simple media switch experiment. Like first, you grow your cells in selective media, so media wouldn't contain the amino acid, and then you switch them to non-selective media, so you add the amino acid. And that means that when you do the switch, you stop selecting against the cells that don't have the plasmid, and then you can observe how fluorescence in your population decays after the media switch due to the fact that you get that plasmid loss events occur and you get an increasing fraction of cells without plasmids in your population. And that's actually what you see in the plot on the right here. So this is the average fluorescence over time before and after a media switch. So before the media switch, you get sort of stable average fluorescence in the population at around 300 arbitrary units here. And then when you switch your media, you start getting an exponential decay of the average fluorescence at some rate. And in fact, so you can fit an exponential to that. You'll find that it is an exponential, and you can extract the rate of that exponential, which I called L here. And then usually people would say, okay, this rate L, that is the plasmid loss rate. It's going to tell me how stably that plasmid is maintained and at which rate it is lost when I'm not selecting against cells without plasmids. And that makes actually sense, because you can go further and you can say, I'm going to write down a very simple population dynamics model here, which has here two variables. One is Z of t. So that would be the number of cells that do have the plasmids. And there's the Z0 here, which is the number of cells that do not have the plasmid. So we're growing in non-selective media here. That means that none of these have a growth advantage. So both of the types of cells would be growing with the same rate lambda. And then you can add sort of plasmid loss by saying I'm going to at a rate L turn cells that do have the plasmid into cells that don't have the plasmid. And if you take this simple population dynamics model and you calculate how the fraction of cells that do have the plasmid change after media switch, you'll find that it decays exponentially at rate L, which is in line with the exponential decay of fluorescence that you're going to find in your data. And that is sort of satisfying to, let's say, 99% of biologists, I guess. That's usually where the story stops and where we've understood the plasmid and we've quantified our system. So when I look at this, I wonder, right? So I wonder what this parameter L is. So how did we actually define a plasmid loss rate here? And we actually defined it by sort of phenomenologically making some model and matching it with some data that we've measured here, but I have no mechanistic understanding of what that parameter L is. If I think about it mechanistically, then I know that plasmid loss is something that occurs as a single cell event inside the single cell, and it's not like sort of a population event or population rate. So I have no understanding of what that parameter L is from that sort of model, and that isn't really giving me any mechanistic insights into what's going on. And what's more, I can actually go beyond looking at the average fluorescence that I've plotted on the top right here, because this is actually data that has been measured by flow cytometry. So I can go beyond the average and look at the full fluorescence distribution that I'm getting here. And if you do that, then you're going to find these distributions here. So on the left is the fluorescence distribution that you're going to measure in the growth period in selective media. So that remains stable at that distribution in selective media. And you see that, in fact, the mean of it is not all that informative, right? So that firstly, it has this very large peak at 0. So peak at 0 means those cells don't have any fluorescence whatsoever, which means they don't have the plasmid. So there's actually a lot of cells that don't have the plasmid in your population. And then you get the super heavy tail, and the mean is somewhere sitting here at 300 arbitrary units, which is actually the 300 arbitrary units that you see in the plot on the top right. And then you can do the media switch with this. And what you're going to find is that the size of the peak at 0, that's going to go up and up more because you're starting to get more and more cells that don't have the plasmid. And correspondingly, the tail of the distribution is going to go down more and more, which is shown on the right here with time flowing from blue to red. And I haven't included the increasing peak at 0 here for scaling reasons of the plot. So looking at this data tells us first two things, right? So the first thing it tells us is, okay, the mean does not paint a full picture of what's going on with the full distribution, which is sort of the classical motivation for doing stochastic models and looking at single cell data and looking at more than the mean. So that's classic. It's still important, but not really the key of what I want to focus on here, which brings me to the second point. Because like when Chetan constructed or started using that plasmid, the first thing I asked him as a person who's trying to do a model of these things is like, what's the copy number of your plasmid, right? And this is usually what we ask any biologist when you're dealing with a plasmid, right? And they're going to tell you something like the copy number of this is 4 to 5. Then okay, 4 to 5, they probably mean average over the cell population or something. So okay, I can deal with this. And try to turn it into a more like the modeling that I do into an actual full distribution or something. But then I looked at the data that Chetan was giving me. I came back to him and say, look, in fact, you may have 4 to 5 plasmids on average in your cells, but in fact you have like 20 to 30% of your cells which do not have any plasmid at all. And he came back to me and said, no, I'm selecting against cells that don't have the plasmid. They are not there. I'm selecting against them. And that's when you realize that there's no contradiction here, right? You just need to take into consideration that there are dynamics here occurring all together at the single cell and population scale, right? So plasmid loss is something that occurs at the single cell process level, but it occurs at the same time scale as the selection against cells at the scale of the population. So what that means is, yes, you do select against cells that don't have the plasmid. You remove them from your population, but at the same time you're creating new cells that don't have the plasmid via the plasmid loss process. And these two things they are going to balance each other, such that despite the fact that you're selecting against cells without plasmid, you're always going to have a fraction of your population present without plasmids. And again, this is something that you can only understand or try to hope to capture if you are in a position to write down a model that tracks both the single cell process as well as what's going on inside the population. Which now brings me to a bit more the actual modeling and the math that we're doing with these. So what we're going to need to do is we're going to need to write down models that describe the single cell process, and then we need to think about what we can do with them in order to factor in the everything that happens at the population scale. So at the single cell scale, the thing that we're working with, or the models that we're working with, are just the absolute standard stochastic reaction networks that people have been doing already since some decades now. So I hope many of you are at least remotely familiar with this. So general formulation here, you get a set of capital K chemical reactions with capital L chemical species, and then these chemical species can be turned for molecule numbers of the chemical species can be modified via all sorts of chemical reactions here. And then these new Ks in the reaction here, so the stoichiometric change vectors that tell you how many molecules of all the species are consumed and produced in the reactions. Okay, so this is super standard. At that stage of the modeling, you're still sort of free to choose whether eventually you want to do an ODE model of your reaction network or a stochastic model of the reaction network. For me, it always ends up being stochastic models. So if you choose a stochastic interpretation of the reactions here, then you'd probably go to something like a Gillespie algorithm to simulate the reactions, or you go to write down a chemical mass equation. And again, this is something that I hope that many of you will at least have heard about. So if we do a stochastic interpretation of a reaction network, that means that we're modeling the reaction network with what mathematically is a continuous time Markov chain. With continuous time Markov chain, we can write down the governing equations for the time evolution of the probability distribution, which in math would be called the Kolmogorov equations. In this context, people call this the chemical mass equation. So this here is sort of what you get is for the chemical mass equation. So just very briefly, this small x in the equation, that would be one possible combination of molecule numbers of all the reacting species that can be present in the population. So this would be a vector with all the molecule numbers. And what we want to do with the model is we want to track in time how those molecule numbers change. So in the stochastic setting, that means we want to track the probability, which is the P here, for small x molecules to be present in the system at time t, and then look at the time derivative of that, like how that evolves in time. And this is what the chemical mass equation actually provides you with. So the thing is that what I've written here is just the equation for one particular state of the system, one particular combination of molecule counts, small x. If you want to calculate with these things, you would have to write one such equation for each possible state x. So you get a coupled system of many ODEs that you eventually need to solve here. And that may be difficult, but again, this is not a topic that I'd like to focus on in this talk. I'm just going to assume that we can sort of calculate with the chemical mass equation for this talk. Okay, so the thing that I'd like to focus on here is where the chemical mass equation really falls short, in the sense that it doesn't allow us to possibly doesn't allow us to describe the joint dynamics of the single cell reaction network that we're modeling and what's going on inside populations. Because effectively what we've written down here is just a single cell model, right? It's a single cell process that we're modeling here, and we've written down an equation for probabilities of this single cell process. And in practice, what you'll find actually many people doing with this is they would take some data such as flow cytometry or mRNA fish data or so, and then sort of fit a model by fitting the solution of the chemical mass equation to the FACS data. And you'll also find that, in fact, in some of the past works that I've been doing, and many other people doing that. The thing is, you need to be very careful with this, because effectively the chemical mass equation is a model of the single cell process, and it's not necessarily valid as a model of something that comes out of a growing population, which is what you're going to get if you do experiments with the growing populations and and you measure data such as by flow cytometry. So what we need to do if you want to get the population scale into the picture is we need to lift the single cell model that we just looked at in the form of the chemical mass equation or the reaction network to the context of a growing population. And the way in which I'm going to do this is I'm going to define a model which is tracking the state of the reaction network, the state of the single cell process that we're modeling in every single cell of an entire population of cells. And then I'm going to get a huge model which tracks a lot of different things. And that model is going to be way too difficult to do anything useful with. But still, I'm going to show you that it can be used as a basis for deriving something that is a lot simpler and that we can actually practically work with. But first, I need to define what the full population model is. And for this, the first thing that I need to talk about is what the actual state of the full population model is. So how can I describe the state of my full population in a mindset where I want to track every the reaction network in every single cell of my population? And this is what I'm doing here with this small y thing here. So this y is going to be the state of my full population, and I'm going to write it as a sum of over all the possible states of the single cell reaction network. And then there's this y of x here, which is a bit an abuse of notation here, but it stands for the number of cells in my population whose internal state, so state of the reaction network, is equal to small x. And then I'm going to put a Dirac delta at that x for every such cell in the population. So basically, imagine that the x stands for numbers of a protein in the cells that we're trying to model. Then this y of x would be counting the number of cells with, say, 10 protein molecules present And if we have like 20 such cells, then we're gonna put 20 such Dirac at X that will then summing up. So that defines the state of the full population. The other thing that I need in order to get a model here is I need to think about how events that occur in the population can alter the state of my population. And the first thing that can happen is a chemical reaction can occur in any of the cells of my population. And that's actually the first line in this table here. So the first line is a reaction of type K happening in any cell of the population. And if it happens in a cell that is in state X, that I'm going to update the state of that particular cell by going from X to X plus nu K. And nu K is the stoichiometric change vector that tells us how the molecule numbers change when that reaction fires. And then at the population scale, what that implies is that I'm going to take my population state Y and going to turn it into a new population state Y, to which I then, from which I subtract one Dirac at the state X from which the cell has left, sort of, and I add a new Dirac at state X plus nu K, because that's where this cell has transitioned to. And this occurs at a rate which is determined by the number of cells with internal state X that I have in my population present times the propensity of that reaction. So this is chemical reactions that can change the state of my population, but I can also have population level dynamics that change the state of my population. And that's these two following rows in the table here. So I'll first talk about the cell death events in the population, because that's actually the easier one here to explain. So imagine that a cell whose state of the reaction network is equal to X dies, right? Gets killed or removed from the population. Then, I mean, this is just represented by X going to the empty set here. The important thing is the question of what we do at the population scale. And the population scale, the only thing we need to do is we need to remove one such Dirac at X from our current population state Y, because we've removed the cell with state X, right? And the interesting thing here really is the rate at which this happens. And the rate at which this happens is again dependent on the numbers of cells that are in my population whose state is equal to X and the death rate of those cells. And the important thing here is that this death rate, so that's this F here, may be a function of X. So that may be a function of the state of the reaction network. So imagine, for example, the plasmid copy number example here that I had before. I could, for example, say I'm gonna put a certain death rate for my cells when the state of the plasmid is equal to 0. And I'm gonna put no death rate for the cells when the state of the plasmid is not equal to 0, right? So this is how I can capture the coupling of like a population level event such as cell death with the single cell process by letting the death rate here depend on the state X. And it's sort of the same story for birth events for cells. You get a rate which depends on the number of cells that have state X times a growth rate. And again, here the growth rate may depend on the state of the single cell reaction network. And this can also do things like burden, for example. Say the growth rate of cells depends on how much of a particular protein they produce. You might want to put a growth rate that sort of decreases with increasing amounts of proteins that are present. So all these, like the dependence of this lambda and the F here on the X, on the state of the reaction network, this is what allows you to introduce couplings between the scales into your model. And the other thing, and I think I'll be brief on that because there's a lot of room for debate and discussion on what is best to exactly do here. So you also need to think about, if you get a birth event of a new cell in your population, then how do you choose the state of the reaction network that you assign to the new cell? And there are a lot of things you can think about, like mother to daughter inheritance type of questions that come up here. The only thing, one sort of simple thing that I represented here is basically saying that if a cell that has internal state X divides and gives rise to a new daughter cell, and I'm going to create a new cell in my population which has the exact same internal state as the mother cell. So this is sort of basically perfect inheritance of the state here. Okay, so the thing here is what I've actually defined here is a population level model that tracks everything that occurs in the population. But conceptually, at least like from a math perspective, it's the same type of model as the plain single cell model that we had initially. So it's a continuous Markov chain that counts stuff, right? Only that what it counts now is a little bit more complicated than what the other model counted, the plain single cell model counted before. Nonetheless, I can still do what people do for a normal single cell model. I can write down a master equation that tries to track the time evolution of the probability distribution of the full population model. And I'm just gonna basically skip through this slide. There's no interest in discussing the details of that. The only thing that I want to put into focus is you can write down such a master equation, but the state space of the population model is so complicated and high dimensional that you shouldn't ever try to solve anything with this. So this is just way too big and too high dimensional to be practically useful, at least if you try to directly solve it. But what you can use it for is you can use it for as the basis for deriving simplified quantities here. And in particular, the simplified quantity that I'd like to put into focus here is this, what's defined here as this R of xt. So you should understand that as the expected number of cells in the population whose state of the reaction network is equal to the small x here, right? So if that is, for example, modeling numbers of a protein in my population, then this could be, for example, the expected number of cells in my population who have 10 protein molecules in them, something like this, or who have 10 plasmids, if it's plasmids that we're modeling. And I can get this expectation by summing over all possible states of the full population model, counting the number for each population state, counting the number of cells with internal state equal to x, and multiplying it by the probability for the full population model to be in that state. And this probability, we don't know it because we can't solve the full population master equation thing, but it doesn't matter because we can derive a dynamic equation for this expected population distribution here. And this equation that we're going to get is actually something that's quite simple and fairly tractable, or at least as tractable as a normal master equation is. And in fact, if you look at that, then you're going to find that the first row of the equation here is one-to-one the same as the normal master equation that I introduced in the beginning. And the only thing that's different is that we get this second line, which stems from the fact that we've added the population scale dynamics into the picture here, right? So first line is just changes in our xt due to chemical reactions. Second line is that you might actually increase the frequency of some particular states x in the population because cells that are in these states are growing faster or dying faster or dying slower or something like this. So that is, in a sense, so the key point to point out here is that this equation is no more complicated than the normal master equation, right? So we can deal with this. If we can deal with the normal master equation, we can also deal with this. The only thing that's sort of different from the normal master equation is that this here actually does not preserve mass anymore, right? So the normal master equation is describing a probability distribution that will always sum to 1. And here we're not anymore at the level of a probability distribution. But that's also not surprising. That's totally how it's supposed to be, because the R of xt here is an expected number of cells with a certain state X in the population, and the expected number of cells that should also grow or decrease with growing or decreasing population size. So it's normal that due to the second term here, where we get to increase or decrease the total mass in this equation. So if you don't like that, this doesn't preserve mass, what you can also do is you can normalize the equation by calculating the total population size, which I've called z of t here. If you define a new rho of xt here as the r of xt divided by the total population size, you can derive a new equation which preserves the mass. And this is in fact almost the same as what I showed you before. The first line is the same, the second line is the same. The only thing that we've added is the third line here, which is sort of a nonlinear normalizing term that you get in the equation, which compensates the increase or decrease in the total mass here. So it's not so important whether you work with the normalized or unnormalized version of the equation. The only reason why I wanted to put this is that on this version of the equation, what you can actually do is you can try to look at a specific case, which is the case when the growth rate and death rates of your cells are actually independent of the single cell reaction network. So assume now we go back to a mindset where what occurs at the population scale is completely decoupled from what occurs at the single cell scale. Then you would put the lambda, the growth rate of your cells, to a constant, the death rate of your cells to a constant that doesn't depend on x. And if you do that in this equation, you'll find that the second and third row of the equation will actually cancel each other out. And what you'll be left with is only the first line, which is the normal master equation. And that's a useful result to get, because this implies that we can actually derive the chemical master equation from a complete population model as describing the normalized expected population distribution. And ultimately, that is also the reason why it makes sense that you can take flow cytometry data and fit a stochastic model to it by comparing it with Gillespie or comparing it with the solution of the chemical master equation. But the additional insight that we've gained here now is that we know now that this is only true if there's no coupling between the single cell process and the population level dynamics. Like growth rates do not depend on what we're modeling, which was definitely not the case in all the examples that I've put up in the beginning, right? So then just for the sake of completeness, you can also define a vector form of this equation. I think I'll skip through that. There's nothing particularly important, only that this equation is eventually what we're using to describe our multi-scale sort of coupled single cell and population dynamics here. And it's sort of as a linear part in the equation, which is similar to what you get in a vector form of the chemical master equation. And it has sort of an additional nonlinear normalizing term here. Okay, so I'm not sure I'm running in time, but probably not all that well. So I'll try to get towards a conclusion by linking that back to the model of plasmid copy numbers or plasmids that I started out with. So this is the same slide as what I've been showing you before. So we were at the stage where we sort of had this phenomenological population level model characterizing plasmid loss and stability of a plasmid, and we were a bit dissatisfied with the fact that this doesn't really tell us in detail what is going on, and it doesn't really provide any insights of how we should mechanistically understand this plasmid loss rate here. So what we can do better now that I've introduced this multi-scale modeling framework is we can actually look at this from the perspective of such a multi-scale model. And that is, I'm going to write down a model of plasmid copy number fluctuations. And I'm going to couple it to what's going on inside the growing population. And then I have a multi-scale model that sort of describes everything that's going on, and I can start to look at what I can gain with this. So here I've put the super simple, maybe stupidly simple model of plasmid copy number fluctuations, which is actually a reaction network that's just a birth-death process here, with birth events standing for plasmid replication and death events standing for dilution of plasmids due to cell growth. I actually know that this model is good enough to very precisely explain all this experimental data that I'm showing you here, which is why I'm using it. But that's only because this is actually a very simple plasmid. So this model, I'm not going to claim that this model is sufficient for all sorts of plasmids, or that it's even intelligent to start with. The thing is, it doesn't matter here. The only thing that I want to put into focus is you're going to put some model of single-cell plasmid copy number fluctuations. And the important thing then is that you take into account how this model couples with what is going on at the scale of the population. And the way in which this is coupled here is that in selective media, I'm going to have to take into account that cells that hit the zero plasmid state in my model are going to be removed from my population, like I'm sort of assuming they die here at a certain rate, which is called mu here. And then in this sort of simple case, I'm going to assume that the growth rates of my cells do not depend on how many plasmids they have. This is also, there's also motivation for modifying this part here. In many cases, for example, if you produce a burdensome protein from a plasmid, then the more plasmids your cells have, the more of the burdensome protein you produce. So maybe the slower they grow. But for this here, we're gonna neglect that. So you'll notice that this model is exactly what I've introduced the formalism previously for. There's a single cell stochastic chemical reactions occurring, and there's these population events. So that means I can go through all the steps that I've introduced previously in order to derive this sort of modified master equation that tracks me the normalized expected population distribution here. And this is in fact something that I can match, and with this model I can actually pretty precisely match the experimental data that we're getting here. So that's one thing that we can do with this. We can match the full distributions of the data that we're getting, but we can also do like more. We can gain insights with now a model that really mechanistically represents everything that's going on here in some sense, at least. So that means that we can now assign a meaning to things such as a plasmid loss rate that emerges at the population scale. Or more generally, you can derive emerging population dynamics models from this, right? I could say I'm now going to take my cell population and I'm going to switch it back and forth between selective and non-selective media, or all sorts of things that I might want to be doing in experiments. And I can represent all those switches in my model because everything here is mechanistically there. And I'm can derive how my population dynamics emerge in that. Like what is going to be the growth rate of my population in selective media, or when I switch back and forth and these sorts of things. And also what an emerging plasmid loss rate is going to be when I switch from selective to non-selective media. I'm gonna skip through some of the things here. The only thing that I really want to conclude on here is you can take that single cell model that I've defined, and you can derive a emerging population dynamics model for the fraction of the population that contains, or the number of cells that contain plasmids and the number of cells that do not contain plasmids from the single cell model. And what you're going to get is something that's going to be the same as the phenomenological model that we sort of just threw at it initially, right? So that means basically that what we were doing initially by just putting the two-state deterministic model, it's perfectly fine, right? It makes a lot of sense. You can derive such a model from a mechanistic single-cell perspective. The difference now is that we actually now understand, if we do this derivation, we understand why and how this plasmid loss rate actually comes about from the single-cell model. And I will have to refer you to the publication for more details on that, but you can show that on the single-cell model, this plasmid loss rate is actually the largest negative of the largest eigenvalue of this matrix C, which appears in the single cell model. And yeah, so we did a bunch of other experiments with media switching experiments with these cells, which I'm going to skip. You can find them all in this paper here. So that was published last month. So that's pretty recent. And this paper is largely focused on the math and the methodology, but it does also contain these media switching experiments. And with this, I conclude, just acknowledge the funding from the French National Research Agency, the INRIA. And this I'd like to put into focus. I recently got an ESC, and I'm saying that because that means that for the next 5 years, I'm sitting on a lot of funding. So if anything of the things that I've been presenting did sound of interest to you, and you're looking for a job within the next couple of years, you might be interested in contacting me and see if there are some cool stuff that we could do possible. I also like to thank the people, in particular Chetan, who's a PhD student, basically doing all the experiments here, and Gregory, who is running the lab where all those experiments were carried out, and then Guillaume Ballis is a postdoc who started in my new team at INRIA recently, and is a mathematician who contributed to the mathematics of these models. So thank you for your attention.